Dr. Vibha, just by way of introduction, perhaps, and you can tell me if I am wrong, perhaps one of the most important points for you today is that, in my introduction, is that you are currently working with a government committee on developing policies and so on for genome editing. So if I'm not wrong, if I am, correct me. But please, as soon as you're ready, if we go with your presentation. Thank you.
Thank you, Peter. Thank you for inviting me, and greetings to all from India. Unlike Argentina, we are still in the process of, defining the regulations and the policies for genome editing in plants.
Dr. Vibha, please take control of your presentation.
Yes. Thank you.
Can you please take the control? Click on the button.
Yes, yes, I have taken. Thank you.
No, result you haven't taken the control. You can click in the upper side of Teams.
As I am clicking, the slides are moving.
But we can't see moving.
Can you now see?
No, you have to click take control in the upper side of Teams window. Take control. Okay. Now
it's done?
Now it's working. Please. Sorry.
Thank you so much. Greetings to all from India, and thanks to the organizers for inviting me. As I said, unlike Argentina, India is still in the process of developing the regulatory policies for genome editing in plants, and I would just like to take you through this process and what is the emerging thinking and the drafts which are which look like the approach that is being taken. So there's a lot of excitement among the scientists from both public and private sector, and about the use of this novel technology for meeting different challenges in the agriculture. In Indian agriculture, there's a lot of research which has been initiated through the funding of the leading agencies, and our National Biotechnology Development Strategy is paying a lot of attention to this applications of gene editing in agriculture. There's a plan to have a mission program, and at the same time and other activities, infrastructure, etc., but focus also on formulation of the regulatory guidelines on gene editing. So as the discussions were initiated in the country about how to move forward, we looked at our existing regulatory framework for genetically modified organisms in India. So we have a kind of a rules 1989, which we call them, and these are the rules under which all the genetically modified organisms and products are regulated. As they were made way back in 1989, the rules are very broad in nature. Of course, the understanding at that time was much lesser as we have now, but the rules have not been changed. Instead, from time to time, different guidelines are issued to deal with the stages or different types of organisms. So we have this implementation mechanism where the Ministry of Environment, Forest and Climate Change, which is also the focal point for the Carta-9 Protocol on Biosafety, which is the leading agencies, but it is supported by the Department of Biotechnology under our ministry to implement these rules. The rules are implemented through a kind of a series of committees, out of which the middle ones, the Genetic Engineering Appraisal Committee, what we call, is the apex body, and then we have different statutory bodies, different institutional biosafety committees, and RCGM. Like this, there's an elaborate structure which is there for genetically modified organisms. Why I shared this with you, because I'll be taking these names as I move along for the regulatory framework for gene editing. So the coverage of the rules 1989 in India, there is a one line which was added in our regulation which said all the new gene technologies, in addition to cell hybridization and genetic engineering, will also be covered under the rules 1989. And when we looked at the definition of the gene technology, it meant that the application of gene tech, it seemed to be covering the gene editing as well. So this was a challenge, and based on this, as I said, there was a need for development of new guidelines and also seeking, some kind of an exemption if at all it is agreed upon. So as I mentioned, these rules are usually, dealt with by updating the guidelines from time to time. So the process was initiated by the Department of Biotechnology for the preparation of guidelines on genome editing. They prepared a document, but it covered, all organisms in one document, and it was opened for public comments in January 2020. However, different stakeholders, since it was open to the comments, the one of the various stakeholders shared their opinion. But the key stakeholder for the topic that we are having today for agriculture, that was our National Academy on Agriculture Sciences. They took it very seriously, and they had a detailed, consultation with very, very important stakeholders. And what they said was that this is a technology that India needs, and this is a technology, all the issues like the questions being asked to Professor Dodna about off-target effects and all those things were discussed. And finally, the conclusions which came up was that there needs to be separate guidelines for genome-edited plants because they have to be dealt with differently, and internationally accepted and clear definitions of SDN 1, 2, and 3 are to be included. So we have gone with the adoption of the definitions which were presented in which were sort of presented in OECD and were more or less agreed by the participating countries. And then it was also discussed, recommended that genome editing is a precise and a targeted mutagenesis tool, so it should be exempted from the regulations and risk assessments under Rules 99, and there is a provision for such an exemption. So they also presented like a regulatory pathway where we had these definitions of the three, the three categories of gene editing in plants, and also, the pathway that was given that for SDN 1 and 2, we are only going to have a laboratory and the greenhouse studies. It will not go for, once it is said that it is free from the external DNA, then it would be treated in the same way as the plants of as the products of the traditional breeding. Whereas for SDN3, where we where there's a new insert, it would be treated in the same way as the genetically engineered plant. So this process, as Peter mentioned, that there is a committee which is working, and I've been part of that committee. After the review of the comments by the stakeholders, there was a stakeholder consultation, and a new drafting committee was set up by the Department of Biotechnology, and I'm also a member of that, and a draft guidance has already been prepared for in about 2 to 3 months work that was done. So now in the key points of the draft guidance is that SDN1 and SDN2 categories of genome editing technologies for plants that do not involve or carry exogenous genes, DNA, and are comparable to naturally occurring variants, these would be exempted from rules 7 to 11. But our approving authority is the Ministry of Environment, Forest and Climate Change. So they have been requested to grant such exemptions under the Rule 20 of the Rules 1989. However, recognizing that this technology can be, needs to be regulated at some point, the initial very early phase of the research and development, that is the prior to confirmation of the transgene-free lines, will be regulated, will be sort of, reviewed by our institutional biosafety committees, and for which we already have a guidelines for doing research activities in contained facility, which will be taken care of. So this draft guidance, which has been made by the committee, has already been approved by one of our leading, recombinant DNA advisory committee. It was presented by the chair of the committee to GAC, and the members were asked to give comments. It is in the final review by the GAC, that is the apex regulator in India. So this is where we stand, and I'll be happy to ask any questions. There's a lot of optimism, and subsequent to this, there has been some delay in, the final approval, and various stakeholders are actually, holding a lot of meetings and requesting the government to come out with this guidelines at the earliest, so that there is an enabling environment to take forward this technology in India. Thank you so much.